Good morning, everybody. I want to express my sincere interest in this topic, and I want to thank the organizers for this, especially Dr. Boros and Dr. Shomiai, for inviting me to give this talk. And I think it's a fascinating area of research, and I'm excited to be here to share our research with you to determine what kind of correlation our data has with the field of deuterium depletion. And I think there's definitely a very tight correlation between the observations that we're seeing and the research that has been done with deuterium depletion. The topic of my talk today will be discussing a non-toxic metabolic management approach for cancer. And specifically, I'll be talking about the effects of the ketogenic diet, which is a high-fat, low-carbohydrate diet that enhances fatty acid oxidation in combination with hyperbaric oxygen therapy. It's important to know that the ketogenic diet shifts the physiology, or shifts the metabolic physiology of a person. So my colleague at Ohio State University, Jeff Volek, studies advanced athletes that are consuming a ketogenic diet, and he measures through a number of very precise measurements the oxidation rates of fatty acids in athletes. And he's determined that a person on a ketogenic diet can oxidize 2 grams of fat per minute. And when we do the calculations, that generates within 4 hours, 3 to 4 hours of exercise time, 400 milliliters of deuterium depleted water within the cell, of about 60 to 70 parts per million of deuterium. So the ketogenic diet, it's important to know that it enhances metabolic water production, which is depleted in deuterium. So I want you to know that information, or keep that information in mind as I go through the data that I'm going to show you. I often like to, before I begin, to acknowledge the funding agency that has supported our lab, the Department of Defense, and specifically the Office of Navy Research, that is a branch of the Department of Defense that funds our research on the development and testing of a mitigation strategy for CNS oxygen toxicity seizures. These seizures are experienced by Navy SEAL divers, also known as special operations divers, that use a closed-circuit rebreather. So oxygen is generally a good thing for your body, but it's a drug, and levels too high can overstimulate the brain and trigger a seizure. We're developing tools in our lab to understand how oxygen is a stimulant to the brain and ways to mitigate that. Other areas of interest that we have active research projects in include neurological diseases, including Alzheimer's disease and ALS, rare genetic disorders, including glucose transporter type 1 deficiency syndrome. It's a rare metabolic disorder where there's a impaired glucose metabolism and transport into the brain, Angelman syndrome, a number of rare genetic disorders that even in the presence of a persistent molecular pathology, a metabolic therapy can be used to symptomatically treat these disorders. An example would be glucose transporter deficiency syndrome, where you have a lack of glucose to the brain, but if you're in a state of nutritional ketosis, where ketone bodies are elevated in the blood, these can replace glucose as the primary fuel for brain energy metabolism. So it's actually the only therapy that's used for this rare genetic disorder. Wound healing is something we've shown that this metabolic therapy can enhance the wound healing process by lowering blood glucose and also enhancing fatty acid oxidation, which reduces the fatty acid profile in the blood because the cells are burning it up, and also reduces the inflammatory pathway, specifically the NLRP3 inflammasome, which recently we published in Nature Medicine that this inflammasome is suppressed by ketone bodies, and we're looking into the mechanisms for that. What I want to really focus on today, though, is our research on cancer. But to talk about our research on cancer, I have to discuss how I got interested in this area of research by studying CNS oxygen toxicity. So we use a variety of tools in our lab to study, to look at metabolic pathways and to understand fundamentally how ketone metabolism and hyperbaric oxygen is working at the level of the membrane and the level of the mitochondria. And we're currently doing metabolomics studies. Our approach is really to cast a wide net and to do global metabolomic studies to determine, looking at 4,000 different metabolites, we determine what metabolites are elevated and what metabolites are deficient for a disease process or a particular metabolic therapy. And that gives us targets to do more elaborate targeted metabolomics, to give us insight into the specific pathology and how the therapies are working. We also do bioluminescent imaging of tumors, and we also we look at reactive oxygen species production in tissue with bioluminescent imaging. I'll just run through these briefly, but some of these you may be familiar with. Atomic force microscopy is a form of microscopy that gives you the scanning, the resolution of a scanning electron microscope, but you can image living tissues with this, or living cells. And it gives you sub-nanometer resolution, and it also, the tip of an AFM interacts with the membrane to give you information like viscoelastic properties of the membrane and how that changes as a function of oxygen or a function of the metabolites in the cell. Fluorescence microscopy, we can image reactive oxygen species production in real time. Confocal, laser scanning confocal microscopy, where we can optically section a specific cell and look at the mitochondria and look at the oxygen-dependent production of reactive oxygen species in the mitochondria and look at mitochondrial membrane potential and correlate that with the cell's bioenergetic capacity or the resting membrane potential. Electrophysiology, I was trained as a patch clamp electrophysiologist. My background is neuroscience and the neural control of autonomic regulation. So how the brain controls our physiology. And we use a lot of electrophysiological techniques to understand this. And radiotelemetry. So we can implant EEGs, diaphragmatic EMGs, EKGs in an animal and look at brain activity, respiration, heart rate, body temperature, these things, and we do it with telemetry. And what we've really done is we've adapted these techniques for use in hyperbaric chambers. For example, here's a study that we've published in the journal Microscopy where we've installed an atomic force microscope and a scanning laser confocal microscope inside a hyperbaric chamber. So we're able to essentially dive the cells, or we do tissues, and in other chambers we do whole animal models. And we look at the effects of oxygen on reactive oxygen species, membrane potential, electrophysiological properties, and to fully understand how these things are working. So an atomic force microscope doesn't use light or electrons. It uses a scanning probe that has a very sharp tip. This tip is, it's so sharp, some of the tips are monoatomic. So it comes down to the point of one atom. And it raster scans across the surface of the membrane to detect subtle changes in the membrane fluidity and the proteins that are in the membrane. And it gives you a very high resolution scan of the surface topography. It also gives you information on the physical properties of the lipids and the physical properties of the proteins. Now jumping to the other end of the spectrum, we do whole animal studies where we implant an electrode and look at, like I said, seizure activity. So this is just a snapshot of some of the tools that we have at the Hyperbaric Biomedical Research Lab at the University of South Florida. So using these tools, we've published a number of papers, and we're putting together a model of, that describes a metabolic hypothesis for CNS oxygen toxicity seizures. So in the process of putting together this model and understanding the molecular and biochemical pathways, we observed some interesting phenomenon in cancer cells. So the information that I'm going to show you in the next 2 or 3 slides will lead to the cancer work. But I want to sort of explain to you, and many of you know some of the pathways that I'm going to be talking about, how this is tied in with cancer. So we know that hyperoxia, or a relative increase in the tissue PO2, or the oxygen concentration, stimulates the production of superoxide anion in the cell, specifically the mitochondria. And also NADPH oxidase is also a site of superoxide production. So superoxide anion is really thought of as a precursor to more reactive species, including the hydroxyl radical, and can also be, in the presence of nitric oxide, can form peroxynitrate. These things can stimulate and elevate lipid peroxidation, protein oxidation, nitration, and cause membrane damage. So we've shown in the brain, specifically in the hippocampus, the CA1 region of the hippocampus, as we elevate oxygen levels, this is the production of reactive oxygen species, superoxide anion, and that increases as a function of oxygen. So superoxide anion and oxygen is a stimulant to the brain. So an overproduction causes cell death and hyperexcitability that triggers a seizure. And we can measure, using scanning laser confocal microscopy, the punctate staining in the cell is the production of superoxide anion in the cells, in the mitochondria itself. So as mentioned, the superoxide can lead to more reactive intermediates that cause membrane lipid peroxidation, and we can use scanning electron microscope or the atomic force microscopy to scan the surface of the cell. And this is just a 1 micron image right here. So it's just cells are about 20 to 30 microns. So it's just a small snapshot of the top of the cell. So these changes, these fundamental changes in the cell physiology, including the membrane lipid peroxidation, alter the electrical properties of the cell, including the resting membrane potential and the action potential firing frequency of the cell, to also lead to an increase in intracellular calcium production. And this can trigger hyperexcitability in the cell. This is a recording from the hippocampus where we measure the orthodromic population spike in response to hyperbaric oxygen therapy. So this is all the neurons firing in synchrony in a hippocampal brain slice preparation. And what we see with under hyperbaric oxygen conditions, we see an increase in the orthodromic population spike and the presence of these secondary spikes. These secondary spikes are analogous to a seizure in a slice. So, and we can measure seizures using our radiotelemetry system inside the hyperbaric chamber. So all this information in this pathway is really leading me to talk about a metabolic therapy that is ultimately going to be used for cancer. But in our studies, to determine how we can mitigate this pathway, we've used antioxidants, anti-epileptic drugs, and oxidative preconditioning, where you expose the animal to hyperbaric oxygen below the threshold for a seizure, and you do it several times to upregulate endogenous antioxidant pathways, hoping that you'll confer protection against a subsequent oxidative challenge that would typically produce a seizure. We've done studies to do this, and we have not shown that oxidative preconditioning, using our protocols yet, protects the animal from hyperbaric oxygen toxicity. But in the literature, in the military literature, I found some interesting observations that starvation ketosis, so taking the animal off a diet for 24 to 36 hours, confers protection against hyperbaric oxygen that would typically cause a seizure. So there was good data supporting that starvation ketosis. So in the absence of food, or glucose specifically, our body mobilizes fatty acids for energy. So the brain cannot readily use fatty acids for energy, so it converts these fatty acids. It might be my phone. So it converts these fatty acids to water-soluble fat molecules called ketone bodies. And the ketone bodies can then replace glucose as the primary fuel for brain energy metabolism. Nutritional ketosis is a way to mimic physiologically the effects of ketosis, starvation ketosis, without restricting calories. And I'll talk more about this, how it's a high-fat diet that can mimic the metabolic effects of starvation ketosis. So what's important to understand is that ketosis, starvation ketosis, and nutritional ketosis enhances beta oxidation of fatty acids. It also, by virtue of the metabolic qualities of ketone bodies, beta-hydroxybutyrate and acetoacetate, it enhances the cell's bioenergetics. So work done by Hans Krebs and his student Richard Veach, who was a mentor of mine as I got into this program, or got into this area of research, he showed that ketone bodies can enhance the delta G of ATP production compared to glucose. So what that essentially means is that per carbon molecule, you can derive more ATP from a ketone metabolite than you can from glucose. So this can enhance, for example, the hydraulic efficiency of a heart. So if you replace glucose with ketones and you look at the perfused heart preparation, the hydraulic efficiency of the heart improves 25 to 30% running off ketones relative to glucose. So we can say that there's enhanced bioenergetics of the cell. And I think most importantly for this meeting is that there's an enhancement of metabolic water production, which is deuterium depleted water. And like I mentioned, with athletes that are oxidizing 2 grams of fat per minute, these are athletes that are adapted to a ketogenic diet. They're oxidizing 2 grams of fat per minute. And if you do the calculation over 3.5 hours, that leads to 400 milliliters of metabolic water, which is depleted in deuterium. So we think that this water, I'm interested in looking at genomic stability in response to the ketogenic diet, and we think that, that the production of metabolic water through ketone and fatty acid metabolism is stabilizing the nuclear genome and perhaps suppressing some of the oncogenes that are driving cancer growth.